morning. I am really excited to kick off what promises to be again this year, strangled up here, again this year a very exciting symposium. We're in store for some really interesting and exciting work today. So what I'd like to do is fill you in on, give you a brief update on some of the work that we've been doing with respect to a relatively novel potential function for Sigma-2, and that is the regulation of metabolism in cells. Can everybody see that? It's a little bit difficult to see. Okay, so as you know, we've been working with Chris McCurdy's group in developing some novel ligands based on this compound, SN79, which is a putative Sigma-2 receptor antagonist. As you can see, the binding data is here. It's not terribly selective for Sigma-2, and we initially set out to try and improve the selectivity of this compound in the hopes of developing a more selective Sigma-2 receptor antagonist. And in the process of working with this group of compounds, we came across this ligand here, CM764, which has an amino substitution on the phenylfluoral ring. And you see we got a little bit improvement in the Sigma-2 selectivity, but that wasn't the most interesting thing about this compound. The most interesting thing about this compound is that it turns out to stimulate some of the hallmarks of glycolytic metabolism. Now, most of the compounds that we had been working with turn out to activate apoptotic pathways in cells. But this one turned out to be different, and we were sort of surprised about this. We measure cell viability by the MTT assay, which just is a general measure of cellular metabolism. It's basically used to determine the number of viable cells you have in your well. And what Hillary observed, and this is work from Hillary Nicholson, what she observed is that there's a dose-dependent increase in MTT reduction. And this occurs without an increase in cell proliferation. So there's no change in the number of cells, even over a period of 4 days. And so what we were actually just measuring is an increase in MTT reduction. And noticing that that's a fairly strange result, I always tell my students, first assume that it is an artifact and then try to prove that it's not an artifact. And so what she did was able to show that this effect is blocked by a number of sigma receptor antagonists. So here you have stimulation of metabolism by CM764 alone. And all of these compounds are able to block this stimulation of metabolism without themselves causing a stimulation of metabolism. And you see here a number of antagonists, BD1047, NE100, BD1063. And SN79 itself is unable to stimulate metabolism, but is able to block the stimulation of metabolism by CM764. This compound, CM572, at higher doses is cytotoxic, but it is a highly selective Sigma-2 receptor ligand, and which we found that in low doses, this compound binds irreversibly to the receptors, and it acts as an antagonist. So we characterize it as a partial agonist. This compound produces a calcium signal that is a transient calcium signal in the cytosol. But we found that it increases the level of ATP in cells. It dramatically reduces basal ROS levels in cells. It's much more effective than alpha tocopherol, in fact. So it's reducing basal ROS levels. But this was the most exciting finding in that there's a time-dependent increase in the stabilization of HIF-1 alpha. And as you know, HIF-1 alpha is sort of a global regulator of metabolism in cells, particularly in cancer cells. And this ligand is able to produce a time-dependent stabilization. And as you know, work on HIF-1 alpha was awarded the Nobel Prize a couple weeks ago. And Bill Kalin was one of the recipients, and Hillary is now working in his lab. Very exciting for Hillary. So Hillary went on to characterize other members of this family, and when we started looking at a number of analogs of SN79, what we found was that, and what we did was we varied the X substituent in the heterocycle and the substituent at the 6th position here. And what we found was that when R is isothiocyanate, regardless of what the heterocycle is, that these compounds all bind irreversibly to the receptor. And what we incorporated the isothiocyanate in hopes of achieving selective irreversible binding to the Sigma-2 receptor, and that is what we found in her initial paper, CM572, is an irreversible, highly selective binder to the Sigma-2 receptor. And all of those compounds with isothiocyanate induce programmed cell death. If the R substituent was anything else, either nitro, amino, methyl ketone, or fluoro, regardless of the heterocycle, these compounds, most of them stimulated metabolism. So there was this divergence in the effect of these ligands. And so what we wanted to do is further characterize the pharmacology of this effect using additional ligands that came out of this study. And the ligands, the additional ligands were CM571 and WA504, structures shown here. And these are their Ki values. They have good affinity at Sigma-2, but they're not selective for Sigma-2. So these compounds all stimulate MTT reduction, so they're all consistent with what we had seen with CM764. And I had shown you earlier that these compounds, that this effect is blocked by a number of Sigma antagonists generally, and then this highly selective Sigma-2 antagonist. But we wanted to look at some additional compounds in this series just to ask some other questions. So this is a related compound, structurally related compound that lacks good affinity at either Sigma-2 or Sigma-1, and these binding values are as defined by tritiated DTG binding. You can see that this compound, which lacks affinity at either receptor, fails to stimulate MTT reduction. Another thing we wanted to look at is these compounds are not very selective for Sigma-2. They have significant Sigma-1 affinity, as you can see here. So we wanted to know, well, what's the possible role of Sigma-1 in this? Now, we knew that plus pentazocine, which is a Sigma-1 agonist, does not stimulate MTT reduction. And Sigma-2 antagonists, Sigma-1 antagonists like NE100 and BD1047 also don't stimulate metabolism. But we wanted to look at this a little further. As you notice here, these antagonists are usually considered to be Sigma-1 antagonists. They lack terrible selectivity for Sigma-1. They turn out also to be Sigma-2 antagonists, but we wanted to look at this a little bit further. This compound here sort of told us that it is selective for Sigma-2, but we wanted to look a little more closely. So here you have a compound in this series which has reasonable Sigma-2 but lacks good Sigma-1. And this compound retains its ability to stimulate MTT reduction. So that's consistent with Sigma-1 receptors not being involved. And another thing we did was we took fragments of active compounds. So the active compounds are basically composed of these two fragments linked by a 4-carbon linker. And neither fragment has Sigma-2 affinity, and this fragment has some weak Sigma-1 affinity, but the heterocyclic fragment has no Sigma-1 affinity, and neither of these compounds is able to stimulate MTT reduction. So that's all consistent with the need to have Sigma-2 activity. So in terms of the pharmacology, using additional compounds, this is all consistent with being mediated by Sigma-2 receptor pharmacology, at least as defined by competition with tritiated DTG. There's still the huge discrepancy between the potencies of these compounds and the binding affinities, which are in the nanomolar range, and then the potencies are all in the micromolar range, which could indicate that there are other proteins being involved. whose pharmacology is simply predictive by the ability to displace DTG. And I'll say a little bit more about that later. Now, another thing we wanted to look into, we wanted to sort of look at the mechanism. And we haven't gone deeply into this, but one of the first things that we did was we wanted to ask, what's the time course of the development of this metabolic effect? And so here what you see is CM764 treating SK and SH cells for various time points to determine when this effect shows up. And you see here that with 1 hour of treatment with CM764, there's no ability to stimulate MTT reduction. If you go to 3 hours, you start to see some reduction, some stimulation of reduction, but it's very variable and not very robust. By 6 hours, you now have a robust and stable ability of the compound to stimulate MTT reduction, and that is almost maximal because we don't see much more if we go out to 24 hours of treatment. So there's a time dependence in the development of this effect. And that time dependence actually correlates somewhat with the time course for the development of HIF-1 alpha stabilization. So Hillary showed that you see a little bit of HIF stabilization at 6. By 12, you have considerably more, and it's pretty stable by 24 hours. So somewhere between 6 and 12 hours, you have this stabilization of HIF. And then it corresponds with later, by 24 hours, the production of VEGF. And VEGF is a target gene for HIF-1 alpha, and VEGF is an angiogenic factor. The other thing we wanted to investigate was the calcium signal, because all of these compounds produce a calcium signal, and we had for a long time known that the calcium signal is not part of the mechanism for cytotoxicity. And so here what we see is that looking at CM764 and 571, there's a dose-dependent release of calcium. These are Fura-2-loaded cells looking at cytosolic calcium. That's 10 micromolar and 30 micromolar of the compound. But what this is showing additionally is that if you pre-treat the cells with dapsogardin to empty the ER stores and then stimulate the cells with the CM764, you completely eliminate that calcium signal. So that calcium is coming from the ER. We don't know what the mechanism is. It doesn't appear to involve IP3, but we're in the process of trying to figure out how this calcium is being released from the ER. And this is just showing that WA504 also is able to release calcium from the ER. Now, what is the role of this calcium? Well, there's literature saying that HIF-1 alpha stabilization is related to these types of calcium signals. So both the calcium signals being involved in the initiation of HIF-1 alpha stabilization, as well as HIF-1 alpha being involved in regulating those proteins that cause cellular handling of calcium. So it could be that HIF is playing a central role in, that this calcium signal is somehow playing a central role in the ability of HIF to become stabilized, and then that downstream is leading to this metabolic effect. So does any old calcium signal then cause the stimulation of MTT reduction? So these cells express muscarinic cholinergic receptors. Carbachol gives a very robust calcium signal in these cells, but carbachol fails to stimulate MTT reduction. So you need to have a molecule that's binding to the sigma receptor in order to get this stimulation of MTT reduction if the calcium signal is in fact related to HIF stabilization and this metabolic effect. You need to have sigma binding affinity. Now, we wanted to look at whether Sigma-2 TMM97 is involved, and we're in the process of CRISPRing out TMM97 and PGR-MC1 in these cells. We don't have that data now, so maybe next year I'll be able to present to you what's happening with that. But there is some interesting data that surrounds TMEM97. Now, if you remember, Hillary had shown that these compounds that bind irreversibly to the receptor that has this isothiocyanate group cause programmed cell death, and then these other substituents produce compounds that have this metabolic stimulatory effect. And last year at this symposium, I talked about the comparison of these monovalent and bivalent Sigma-2 ligands. So here you have CM571 and CM572, which is the isothiocyanate compound that Hillary showed is cytotoxic. And I talked last time about this compound, MAM03055A, which is really a dimer, if you will, of CM571. And we compared the ability of these cells to affect cell viability. You can notice that MAM is very selective for Sigma-2 over Sigma-1, so it's about 60-fold selective. And the surprise was that MAM, as opposed to being similar to 571, is actually more similar to CM572. So MAM is cytotoxic, and this decrease in MTT reduction is actually cell death. MAM induces an apoptotic cell death with bid cleavage and other downstream markers that we had observed with other cytotoxic Sigma-2 ligands. And the other surprise was that MAM, like CM572, doesn't come off of the receptor. It binds pseudo-irreversibly. Now, there's not a moiety in the molecule that can react covalently with the receptor like there is with the isothiocyanate. But for some reason, it does not wash out of the receptor very readily. It may have a very, very slow dissociation rate. But CM571 binds reversibly to the receptor and stimulates metabolism. So these two irreversibly binding ligands are cytotoxic, and this one is metabolic stimulative. And when we look at what these compounds do to TMEM97, what we find is that CM571 has no effect on the levels of TMEM97. This is just treating the cells 30 micromolar for these various time periods, and then measuring TMEM97 in a cell extract by Western blotting. But MAM produced this time-dependent loss of TMEM97. So this irreversibly or pseudo-irreversibly binding ligand causes this time-dependent loss of TMEM97. Now, this may be related to the data from Bob's lab showing that you'll hear more about later, that there's an internalization of TMEM97 along with PGR-MC1 and the LDL receptor. We don't really know what this means, but it could be explaining or giving some explanation for why these two classes of ligands may be different. So we have this one is cytotoxic, this one is metabolic stimulative. We don't know what the relationship is, and it's going to require further study. So in summary, the metabolic stimulative effect of SN79 analogs has clear Sigma-2 receptor-like profile, with the exception of the discrepancy in binding affinity, at least as characterized with DTG. The time course for the development of the metabolic effect is consistent with the time course for stabilization of HIF1, and the Sigma-2 ligands that are metabolic stimulators induce a transient calcium signal from stores in the ER. And those types of signals have been linked to HIF expression, and the non-sigma calcium releaser failed to produce the metabolic stimulation. Now, HIF is involved in the adaptation of cells to hypoxia. So HIF affects angiogenesis and blood supply. It also affects glycolytic metabolism. And Sigma-2 receptor and HIF could be potential regulators of the Warburg effect, which is how cancer cells have adapted to living in hypoxic environments. So the Sigma-2 receptor may cause an increase in calcium, an induction of HIF by some mechanism, and we've shown that upregulates VEGF, which gets you over here to increasing blood supply. We're now in the process of getting more concrete readouts for metabolism other than just looking at MTT reduction. So we're doing some seahorse experiments. Hopefully I'll be able to talk about those next time. But the preliminary data that we have does suggest that there's an increase in lactate extrusion from the cells with measuring the ECAR in the seahorse apparatus. And hopefully I'll be able to talk to you about that more next time. So the possible implications are, in cancer, this is consistent with upregulation. It could be involved in Warburg glycolysis and enhancing tumor angiogenesis via these VEGF secretion. And it'd be interesting to see what this effect looks like in neurons, which could be production from oxidative stress with that reduction in ROS levels, protection from hypoxic stress by the upregulation of HIF, and this could have implications for these types of compounds in stroke and neurodegeneration. The divergent effects of these analogs could involve effects on TMEM97 levels. As I showed you, the irreversibly binding analogs induce cell death and cause a loss of TMEM97. The reversibly binding metabolic inhibitors have no effect on TMEM97 levels. Alternatively, these effects could be mediated by different classes of Sigma-2 like proteins or different binding partners such as PGRMC1. And what we really need are better radioligands so that we can fish out these two effects. It could be that what's happening is that DTG binding, which is a good ligand for TMEM97, may simply be predicting the pharmacology of related proteins that don't bind DTG. And so we could be seeing overlapping pharmacology with these compounds acting through compounds that just don't bind DTG, but they have such a similar pharmacological pattern that they look like they're Sigma-2 mediated effects. That's just a hypothesis, and we need more work to figure that one out. So acknowledgments. So most of the work that I showed you was by Bridget McVeigh, who was a master's student in our lab. Sherry Liu did some of the work, and then Hillary's work was also cited. And then these are our chemistry collaborators, Chris McCurdy, Christophe Massanju, and Marco Martinelli, and then this is the funding. Okay. Take questions.
Very interesting work. I wanted to ask you, and I think you kind of were addressing it, that you're going to take this into the seahorse to see whether or not you look at mitochondrial function. I would maybe like to put in a plug to maybe look at mitochondrial fission-fusion dynamics.
Sure.
The reason is some years ago, I think thapsigargin's been shown to actually influence mitochondrial fragmentation, and that could potentially influence the data, to say nothing of the fact that MTT reduction is a product of mitochondrial activity. So there may be an opportunity to perhaps distinguish between the endoplasmic reticulum as the source of calcium versus the mitochondria.
Versus the mitochondria, right.
But it just struck me as that, and I thought I'd share that.
Right. So my understanding is that MTT reduction occurs both in the cytosol and in the mitochondria by NADPH and NADH-dependent reductases. And it's interesting that the compounds in this series and other series that we've looked at, actually the cytotoxic compounds, do so by causing mitochondrial depolarization. So all of this could have something to do with regulating how mitochondria function. I'll give you a look at it. And we do have the preliminary data that we have with the seahorse does suggest that there are alterations in ECAR, but we don't have, I didn't want to show that data because we haven't yet optimized the conditions for that, but hopefully next time we'll be able to talk about that.
Thanks, Wayne. I'm wondering if some of your compounds that are acting through different mechanisms there, can you tease apart the calcium and the mitochondrial effects on HIF? So I'm just wondering if some of the compounds that are stimulating mitochondrial activity are actually causing a very localized hypoxia, and that's leading to the HIF, or if there's some more direct
It's
activity.
possible because we see that dramatic drop in ROS, right? So there could be sort of a feedback mechanism that is related to that. Where calcium comes in, calcium has been shown to be upstream of HIF and also downstream of HIF. So HIF can regulate calcium dynamics as well. But yeah, any of those mechanisms might be coming into play.
Okay, last question. So you mentioned the time-dependent loss of TMM97 that could potentially be due to internalization. Did you have
we
any?
don't have any evidence that just feeding on what has come out of Bob's lab
Right.
with the internalization of the complex. This could be what we're seeing. I don't know if we're going to hear more about what happens to the TMM after it gets internalized. Does it get degraded or does it come back to the cell surface?
Right.
We don't know. But this data was just total cell extract, right, from cells that had been treated. So I don't know whether that's degradation or
Right.
or turnover, increase of turnover. We don't know yet.
All righty. Okay, I think we need to move on to our next speaker. So our next speaker is attending via video feed, and this is Marti Colomcadina from Dr. Tara Spires-Jones' lab. And there he is. Wave, Marti. Can you hear us? All right.
Yeah, I can hear you. Can you hear me?
We can, and the sound quality is excellent. So, so Aileen is putting your slides up. Just give us a second. Let's go full screen.
Ryan's coming down. Oh, there we go.